Hello students, welcome to Vivid Botany. We are discussing the NEET questions of 2020. This question is about the biological nitrogen fixation, the enzyme nitrogenase. We know that nitrogenase is nowadays called as dinitrogenase, and this dinitrogenase is exclusively present in some prokaryotes. It is not present in all prokaryotes. It is present in some prokaryotes. They are called as diazotrophs. Like we have a big list: Rhizobium, Bacillus, Clostridium, Azotobacter, Rhodospirillum, Anabina, Nostoc, many more. So they are called as here diazotrophs. The question arises here that the enzyme dinitrogenase is of the plant or bacteria. This dinitrogenase is exclusively belong to the bacteria only, because the evidence says that the nitrogen fixing genes, that is the genes encoding dinitrogenase enzyme, are present with bacteria only. That is, the genes encoding nitrogenase are present with bacteria, that is rhizobium. One example that we can write is that rhizobium. So let us see that there are two types of genes are there. The genes encoding nitrogenase are called as NIF genes, nitrogen fixation genes. Another is the NOD genes. They are called NOD genes and NIF genes. So if you see the reaction of Habers process, I would like to quote the Habers process reaction which you find in chemistry, that is N2 plus 3H2 gives rise to 2NH3, this ammonia. So here the iron is the catalyst, whereas the molybdenum is promoter. So here there is no extra hydrogen that is used in reactants. Exactly 3 hydrogen molecules are being utilized to form 2 ammonia molecules. So completely this is balanced reaction. The copy of dinitrogenase, biological nitrogen fixation, that takes place in diazotrophs is the Habers process. Habers process is the copy of biological nitrogen fixation. So if you see the biological nitrogen fixation reaction, here we take dinitrogen. We know dinitrogen is diatomic molecule. It is molecular nitrogen. One nitrogen is having 3 bonds with another nitrogen, that is one sigma bond and 2 pi bonds are present in between the nitrogens. So this breaking of this bond is very difficult, and bond energy is very much required to fix this nitrogen. So dinitrogen plus 8 protons plus 8 electrons plus 16 ATP. The metabolic energy required here is 16 ATP to give rise to 2 molecules of ammonia. Remember one thing, that one hydrogen is equal to 1 electron plus 1 proton. One hydrogen is equal to 1 electron and 1 proton. Here we have taken here 8 electrons and 8 protons. So 8 electrons and 8 protons is equal to 8 hydrogens. 8 hydrogens is equal to, we know that is 4 hydrogen molecules. But in case of the ammonia, the number of hydrogens are only 2 3s are 6, that is 3H2, or we call it as 6H, are required for formation of 2 ammonia molecules. Then what about the rest of the 2 hydrogens? Yes, we are coming to that point where the other byproduct is here H2. This is the other byproduct, right? Next we have here 16 ADP and 16 inorganic phosphates. So the byproduct includes here hydrogen, ADP, inorganic 16 inorganic phosphates are required here. So the answer is here is that during the biological nitrogen fixation by nitrogenase, here ammonia is not alone formed, but along with ammonia, hydrogen is also formed. So answer is here 4. And one more thing here is that for 2 ammonia molecules, 16 ATP are required, whereas for 1 ammonia molecule, the amount of that are required includes here 8 ATP, along with that plus 4 electrons and 4 protons are required. Then why should we take here 2 nitrogen atoms? Remember that in the dinitrogenase enzyme, if you see, for suppose this enzyme will have 2 active sites. So in each active site it can bind with 1 nitrogen, so that it can bind with 2 nitrogen atoms at a time. It cannot bind with only 1 nitrogen atom, it can bind with 2 nitrogen atoms, and it is added with hydrogen molecules one after the other like this. H2, H2 and H2. Like this they are added 4 times. In fact they are added only 3 times, and it forms here 2 molecules of ammonia that are formed. So because of this reason, as the enzyme has got 2 active sites, so it can bind with 2 nitrogen atoms at a time. So this is the answer here, that apart from the ammonia, hydrogen molecule is also formed and 16 ATP are required. So it is an active process. Do remember that it takes place all biological nitrogen fixation process. So I think the equation is helpful for you. You have to practice the equation again and again to get it thorough. Then only you can know for 1 ammonia molecule 8 ATP are required, for 2 ammonia molecules 16 ATP are required. And as well as sometimes they may ask here that the ratio of number of protons, electrons and ATP, that is number of protons, number of electrons and ATP ratio will be something like that 1:2. This is the ratio required for fixation of 2 molecules of biological nitrogen fixation. So please subscribe my channel, that is Vivid Botany, and make a comment also that whatever you did not understand, if you drop a line in the comment box, we will be looking towards that in our next video. So thank you very much.